Assalamualaikum and hello everyone. In this presentation, we will discuss two parts of our assignment, Part A and Part B. For Part A, two hydrocarbon isomers A and B with the molecular formula C5H10 are separated by GC and subsequently analyzed by NMR. The carbon-13 NMR data are recorded as follows. And for the first question, based on the carbon-13C NMR data, deduce the structure of hydrocarbon A and B. In hydrocarbon A, carbon-13 NMR, we see 5 signals with multiplicity of the peaks. One singlet at 132 ppm represents quaternary carbon, one doublet at 118 ppm shows methyl group, and 3 quartets at 26, 70, and 13 ppm each represent methyl group. So based on this data, the molecule has 1 quaternary carbon, 1 methyl, and 3 methyl group. Next, we calculate the index of hydrogen deficiency for C5H10 molecule to determine the characteristic of molecule structure. And based on this calculation, 1 degree of index hydrogen deficiency means that the molecule has 1 double bond or ring but not both present in molecule structure. We can deduce that the structure for hydrocarbon A is 2-methyl-2-butene based on carbon-13 NMR signals and the index of hydrogen deficiency obtained.
Hello and Assalamualaikum, my name is HUMAIRAH. So I'll present the analysis for hydrocarbon B, which has the molecular formula C5H10. Alright, so based on the equation, based on the carbon-13 NMR data, there are 5 carbon signals and the multiplicities help us to identify the types of carbon. So if you can see that, there is a delta 147 ppm. We can see the singlet, which is represent a quaternary sp2 carbon in an alkene, meaning it's a carbon with no directly attached hydrogen. And at 108 ppm, there's a triplet indicating a CH2 group. This CH2 group is vinyl, meaning it is a directly bonded to a double bond. And another CH2 group appears at 31 ppm as a triplet too. This is a more saturated environment likely to be allylic. And the signals at 22 and also 13 ppm are quartets representing 2 different CH3, which is a metal groups. So from this, we can deduce the structure is likely to be 2-methyl-1-butane, which is a branch alkene with 5 carbon atoms. Alright, so now looking at our IHD, which is the index hydrogen deficiency calculation, is the same as what my friend already told in the previous slide. So the calculation is the same, so we get the value of 1, which means the compound contains 1 double bond. So the structure that fits both molecular formula and also the spectral data is 2-methyl-1-butane. As shown here, we can see the double bond between the first and the second carbon and a metal group attached to the second carbon. So this structure matched perfectly with the NMR signals and the fragmentation pattern that we discussed earlier.
Moving on to the Part A2, predict the major peaks recorded by the mass spectrometry instrument of each of the hydrocarbons. What species do this peak represent? In mass spectrometry, 2-methyl-2-butene has molecular ion peak at mass-to-charge ratio MZ70. It undergoes fragmentation by losing a methyl radical, forming a resonance-stabilized allylic cation with MZ55. This allylic cation gives the most intense signal in the spectrum due to its stability by the localization of positive charge, making MZ55 as the base peak. Further fragmentation caused another loss of methyl radical, forming allyl cation with MZ41. The allyl cation further fragments by losing a methylene radical with MZ14, resulting in the formation of vinyl cation with MZ27. From the table shown, we can see several significant fragmentation peaks along with their corresponding mass-to-charge ratio, MZ. The major peaks are at MZ17, 55, 41, and 27, each representing a different positively charged ion formed by the cleavage of the bonds in 2-methyl-2-butene. So the most intense peaks is at MZ55 due to the stable allylic cation.
So this table summarizes the major peaks detected in the mass spectrum of hydrocarbon B. So each mass-to-charge ratio, or MZ, value corresponds to a specific fragment ion, and the pathway shows how the molecule breaks apart during the ionizations. So as we can see that at MZ70, so this is the molecular ion peak representing the entire molecule. So it confirms that the molar mass of hydrocarbon B is 70 gram per mole. So as we can say that that's the ion formed directly from the molecule. So we can see that fragment pathway. And at that MZ55 allylic ion, so this fragment forms after the loss of a methyl group CH3 from the molecule. So it is a resonance stabilized allylic cation, which is very common in alkane. And then at MZ41, propyl or vinyl ion, so this ion results from a cleavage near the double bond, which produce a stable C3H5 ion cation. So this is a very typical fragment for alkenes. And then we have at MZ29, the ethyl ion. So this is also a very simple alkyl fragment indicating a 2-carbon chain. So it's formed when a part of the molecule cleaves to leave CH3CH2 ion. And at MZ27, vinyl ion, a common small fragment for alkenes. So this ion shows the molecules contains a double bond, which it comes from CH2 double bond to CH ion. So all these fragments support the presence of an alkene functional group a linear or slightly branched hydrocarbon chain, and it confirms the structure deduced from NMR data earlier.
Okay, hi, my name is IRDINA BATRISYIA RASHIDI, and I will be explaining the part B of instrumentation of gas chromatography, isotope ratio mass spectrometry, or called as GCIRMS, which is a powerful combined analytical technique. Okay, so in this slide, we can see the latest type of thermoscientific GCIRMS machine by Thermo Fisher Scientific brand. Okay, so GCIRMS is basically a combination of two systems, which is first, gas chromatography, which separate volatile compounds in a complex sample based on their own volatility and interaction with the stationary phase. And also, isotope ratio mass spectrometry, which is our IRMS, which measures the ratio of stable isotopes like carbon-13 to carbon-12 or nitrogen-15 to nitrogen-14. So together, this combination will allow scientists to analyze isotope ratios of the molecule level, even in complex mixture. Okay, so here's the basic components and the concept on how GCIRMS works. Okay, so first we have sample introduction system, which introduce the sample into the GC oven where it separates individual compounds in a complex mixture. Then we have conversion interface that converts organic compounds into simple gases that is suitable for isotope analysis, either through combustion or pyrolysis. So next we have drying or water removal system that helps to eliminate water vapor from the gas stream to prevent interference. And also we have carrier gas flow system that will allow sample and gases movement through the system under controlled conditions. And next we have the open split interface, which will divert a portion of the gas to the IRMS, while the excess will goes to waste. Okay, and we have reference gas input that will introduce the standard gases for calibration and also quality control. So next also we have the isotope ratio mass spectrometry, our IRMS, that will basically measure the isotope ratios of the target elements in the sample. And lastly, we have the data system that will collect and also process isotope data and control instrument operation. So in this slide, we have the schematic diagram of the GCIRMS instrumentation. Okay, so I will explain the schematic diagram of GCIRMS instrumentation first. So the autosampler will inject the sample into the gas chromatograph, which is our GC oven or GC column. Okay, so in the GC oven, the sample is then vaporized and separated into individual components using a capillary column. So after separation, compounds can optionally be directed to a detector like FID or MS for chemical identification. So for isotope analysis, compounds can continue into the conversion interface. So we have combustion oven or reduction oven. So in combustion oven, organic compounds are oxidized to form CO2 for carbon analysis and NOx for nitrogen analysis at high temperature. And in reduction oven, NOx gases are reduced to N2 gases for nitrogen isotope analysis. Okay, so next, the drying unit. Okay, in drying unit, water vapor generated during combustion is removed using selective membrane dryer to ensure clean gas enters the IRMS system. Okay, so in carrier gas system, helium is used to transport the gases smoothly through the system, and we have reference gas inlet. Okay, so a non-reference gas such as CO2 is introduced periodically for calibration and comparison during isotope ratio measurement. And then we have open split interface. Okay, in this part, a small controlled portion of gas flow is directed to the IRMS while the rest is vented. Okay, so next, the IRMS system. So we have ion source. So ion source, in the ion source, the gas molecule are ionized using an electron beam. And then we have ion optics and accelerated in the IRMS system. So the ion beam is focused and accelerated through lenses for a better resolution. Okay, so next, the ionized ion will move through the magnetic analyzer, which they will be separated by their mass to charge ratio, which is MZ, using a magnetic field. So they will move through the Faraday detectors so that the isotopes are detected based on their MZ values. So we can see the MZ values that is already presented on the slides. Okay, so lastly, the ionized ion will go through the amplifier and data system, which is our computer. So the signal is amplified in the amplifier, and then the data system will calculate the isotope ratios and display the result.
Now, I'm going to explain about the application of GCIRMS. In forensic investigations, GCIRMS serves as a critical tool for source attribution and differentiation of materials that might be chemically similar but originate from different sources. For instance, in drug profiling, two samples of the same illicit drug, for example cocaine or heroin, might have identical chemical compositions, but their stable isotope ratios will vary depending on the geographical origin of the raw materials, the synthetic pathway used, or even the cultivation conditions. This enables forensic scientists to determine if different seizures of a drug share a common batch or source, aiding in supply chain disruption. Similarly, it's used to compare substances like explosives, fibers, paints, inks, tapes, or adhesives to establish whether they share a common source or manufacturing history, thus linking suspects to crime scenes or distinguishing counterfeit products from genuine articles. The method's ability to reveal subtle isotope differences provides irrefutable evidence in legal contexts. Other than that, GCIRMS is indispensable in understanding environmental processes and tracing contaminants. In environmental forensics, it helps identify the source of pollutants by analyzing the isotopic signatures of specific compounds found in contaminated water, soil, or air. For example, it can differentiate between petroleum spills from different origins or identify the source of specific industrial chemicals. This is vital for assigning responsibility for environmental damage. In environmental monitoring, it provides insights into nutrient cycling, carbon sequestration, and greenhouse gas emissions. In wildlife forensics, GCIRMS is used to determine the geographical origin of animal products such as ivory, timber, and fish, which is crucial for combating illegal trade and enforcing conservation laws. These isotropic ratios in an animal's tissue reflect its diet and the isotropic composition of the water it consumes, which are influenced by the local environment. GCIRMS is also useful in food authenticity and traceability. The global food market is increasingly susceptible to fraud, such as mislabeling or adulteration. GCIRMS is a powerful defense against this, primarily by establishing the geographic authenticity of foodstuffs. The stable isotope ratios of carbon, oxygen, and hydrogen in food products like wine, honey, olive oil, or fruit juices are influenced by the local climate, water sources, and agricultural practices of the region where they were produced. For example, C3 and C4 plants have distinct carbon isotope ratios, and water sources vary geographically in their hydrogen and oxygen isotope ratios. By analyzing these isotopic fingerprints, GCIRMS can verify the declined origin of a product and detect if cheaper ingredients have been added, thus ensuring product integrity and protecting consumers. In biological research, GCIRMS provides unique insights into metabolic pathways and ecological interactions. For instance, in human and plant physiology, it's used in metabolic studies to trace the fate of specific compounds within an organism. By feeding subjects with isotopically labeled nutrients, researchers can track how these compounds are metabolized and incorporated into different biomolecules, differentiating between endogenous and exogenous biochemicals. This is particularly relevant in sports medicine for detecting the illicit use of performance-enhancing drugs such as synthetic testosterone versus naturally produced testosterone, or in toxicology. In ecology, GCIRMS is fundamental for studying photosynthetic pathways, unraveling complex food webs, and understanding ecohydrology and nutrient cycling within ecosystems. It is also contributes to human provenancing in archaeology and anthropology by analyzing isotopes in skeletal remains to infer diet and migration patterns. Last but not least, GCIRMS also is very useful in geosciences, archaeology, and paleoclimatology. GCIRMS plays a vital role in understanding Earth's past climate and geological processes. In paleoclimatology, the isotopic ratios of organic molecules preserved in sediments or ice cores can reveal information about ancient temperatures, precipitation patterns, and atmospheric carbon dioxide levels. In geochemistry, it helps in tracing the origin and alteration of organic matter in rocks and sediments, providing insights into petroleum formation and natural gas reservoirs. In archaeology, isotope analysis of human and animal remains, as well as artifacts, can reveal dietary habits, migration patterns, and the origins of raw materials, contributing to our understanding of ancient civilizations and environments.
So that will be all from our group. Thank you.